So you will notice that I'm an organizer and not an official speaker for the meeting. And so I have a task which is quite difficult. You all come from very, very, very different disciplines than biology. Some of you have never seen biology before. You have an interest in it. Others, in fact, some of you have done research and published papers. Okay, so it's a very broad spectrum of participants here. And we are really going to launch very deep into all kinds of biological phenomenology starting tomorrow onwards. So what I am hoping to do between now and lunchtime is to introduce you to a little bit of jargon in biology, some of the fundamental concepts in biology, just so we're all on the same page. And then I did structure my talk like the talks of many other speakers here, in fact, as Rahul alluded to. So it's actually very useful to see these transitions, the interface between physics, quantitative disciplines, and biology by example. So after I finish my general comments, which hopefully then you don't have to read a biology textbook after that, I will give you an example from our own work, which again will introduce to you concepts of genetic networks, which is basically the title of this talk, and also introduce you to the idea of testing these sorts of quantitative predictions experimentally. Okay? Now, the way the school is structured, it's meant to be completely interactive. Okay? We have 1.5 hours now. We have time in the afternoon. In fact, every day there are tutorial slots left over. These are sort of spillover slots. So do not hesitate to stop the speaker and say, I didn't understand what you said, or here's an interesting question, and we can have a discussion about that. Okay? So it's not a series of lectures, it's a school, and you're meant to participate as much as the people on this stage. Okay? So please do not hesitate to ask a question. If you have a question, chances are many others in the room will have a question. Okay? Fine. A little bit about my background. I have a PhD in physics. It was during sort of midway through my PhD that I discovered biology. I was working on basically statistical mechanics, stochastic processes, and we started to apply some of these problems to understanding what's happening inside a cell. And I was lucky enough to have experimental collaborators who really did designed wonderful experiments to test these physical theories. So this was over 10 years ago now, and I've been back at NCBS, which is an institution you heard about earlier. It's just up the road, doing a lot of computational work, but we also have a small lab in which we grow bacteria, modify them, and so on. Okay, so let's get started. So I titled the lecture, The Watchmaker's Apprentice. So what does this mean? So this is a famous book by Richard Dawkins. Richard Dawkins has made it his life's mission to convince people that evolution is real and that nobody should criticize this. You can have some issues with how he sends the message across, but his message is very, very clear, that evolution works like a blind watchmaker. And why does he use this particular metaphor? So he's talking about the following thing. If you walk down the road and somewhere on the road, on the ground, you see a watch, right? It's a watch, it's working, it's telling the time, it's moving, maybe it's making an alarm. The immediate conclusion you draw from that is that there must have been a watchmaker, right? If there is an object that is created, that means there was a creator of that object. And that basically is, so it's in fact the complexity of biology is one of the strongest arguments people called creationists use to say that there must be some unknown force out there which can create objects of such complexity. And what Dawkins says is no. So there is a force, but it's not an unknown force, it's the force of evolution. And what evolution does is over billions of years, it works by tinkering, making small improvements, right, until you go from simpler forms to more complex forms, explaining essentially all the diversity of biology we see today. There is nothing left to explain. So this is actually very well understood, and we will have lectures on evolution tomorrow and day after tomorrow that will explain sort of the quantitative aspects of this wonderful hypothesis. Okay? Now, I'm not going to talk about the blind watchmaker, the force of evolution. I'm going to talk about who we are. So we see ourselves as the watchmaker's apprentice. Okay? So what does that mean? It means I'm working in sort of the warehouse, the shed of this blind watchmaker. And this force over billions of years has made all these intricate parts, these proteins, DNA, all kinds of other aspects of biology, cells and so on. And there's just this treasure trove of all this biological material. So what we can do is just raid the treasure chest and try and make something else out of it. Okay? So we're not trying to recapitulate evolution. We're just saying there's all this wonderful stuff that the watchmaker has made. Let's break it apart. Let's take out all the gears. Let's see how they work. Let's take out the springs. Let's see how they work. And then let's put them together to make something else. Okay? And what else? Well, so that's based on your design. Right? So I have something in mind. I want to make something. and I'm able to take all these pieces, and if I'm lucky, I'm able to make something else using a process that's inspired not so much by evolution, but by engineering. Okay? So I'm going to try and convince you this is difficult, but it's possible. And it's going to happen more and more often as the century progresses. Okay, so first, as I said, I'm going to give you some background. So you know who these people are, Charles Darwin and Gregor Mendel. They were actually contemporaries. Their most famous work that continues to have impact was done roughly the same time in the early to mid 1800s. There's no evidence that these two men actually met each other or read each other's work. Darwin had a huge library, but there is no evidence that there is a rumor, but there's no evidence he was actually able to understand what Mendel had done. But let me tell you first what Darwin did. So Darwin and Wallace at the same time set out to explain the diversity of natural form. All the things that we see today that count as life, which seems to be on the face of it a completely different phenomenon than all the sort of physical objects that we see, including stars and galaxies and atoms and electrons. Something is different. And Darwin put forward this proposal that the way you make all this diversity of life is by gradual changes to existing forms. Okay? So if you look at the ancestor of humans, they didn't look like humans. They looked like maybe some sort of ape, not the ones that are alive today, but the ones that we see in the fossil record. And what Darwin did was he said that based on this hypothesis, the prediction is that life should be, all living things should be related in a tree-like fashion. Right? This is a non-trivial prediction. So before Darwin, you had Linnaean taxonomy, which tried to classify life based on shared characteristics, which is fine, but you also had this very misleading concept called the great chain of being, where the life is organized on a straight line, and you go from simple organisms, bacteria and mold and so on and so forth, not bacteria then, but insects, and then you have worms, insects, and then you go to lobster and so on and so on and so on, and humans are at the pinnacle of this. Right? And Darwin says that's not the case. Biology is actually built like a tree. There are ancestral forms that diversify based on this selective pressure, which he called natural selection, to make all the diversity that you see in the present day. So this is a very, very important concept. Now what Darwin, if you read his book, it's a very easy book to read and it's wonderful to read. He had only a few ingredients to his theory. He said that there are populations of organisms, okay, and when parents have offspring, the offspring are slightly different from the parents. Okay, so the offspring share a lot of the characteristics of the parents. So there's this idea of inheritance, but the inheritance is not perfect. Okay, so if you allow this imperfect inheritance, you then generate diversity of organisms in a space and time. And then some of them will die and others will not, because there's a sort of brutish competition for natural resources. Right? And the ones that survive that process are the ones that propagate their genes to the next generation. Okay? And this was his theory, and that explains a huge amount of data if you read his book. What Darwin did not do, and he was well aware of the problem, but he chose not to address it. He said, I don't know how it is that that a parent can have an offspring, and the offspring somehow contains the template to make something which is an identical or a near identical copy of the parent. Okay? He did not want to speculate on the mechanics of inheritance. He said it exists, it's clear that it must be happening, and that explains all the rest of it. Now, what Mendel did was completely complementary. So he was actually doing experiments on plants, and you've all studied this in school, so I'm not going to belabor it. But basically Mendel's discovery was that traits can be inherited. So he was looking at traits like color and height, size of plants and so on, but even more complicated things. Traits can be inherited. And the mathematics of this inheritance seems to imply that these traits behave as small particles that are inside an organism, and they get passed on and reassorted in a combinatorial fashion to give the offspring with similar traits. Okay? So this is actually a very important idea. If Darwin had read this at all, he would have completely put everything together. Okay? So these traits behave as particles, and those particles are what biologists call genes. It's an abstract concept. It's a particle that's inside me somehow, and if I have offspring, it will be copied and transferred to the offspring and then define the traits of the offspring. So again, it's just a way to explain the quantitative facts. That's all it was. Okay. So not long after, you see late 1800s, and now you have the mid-1900s. So you've already seen this is Erwin Schrödinger. Exactly 70 years ago in Trinity College, Dublin, Schrödinger gave a lecture, which was later expanded into this book called What is Life, which is freely available on the internet and you can read it. So Schrödinger addressed this question of how it could possibly be. So he read about the biological phenomena and he said, well, so it clearly appears to be that there's some sort of particle, and he reinterprets it in the context of mid-20th century science to say this must be a molecule of some kind, right? But a molecule with very special characteristics. If you try and explain how reproducible patterns occur in physics, right? Usually you use the law of averages. In other words, large numbers of samples. Statistically, things behave very regularly. Instances of a statistical distribution do not behave regularly. Okay, so the average height of people is slowly varying over the centuries, but because the height of all of you is very, very different from the average. And he was puzzled, how, all of statistical mechanics and quantum mechanics tells you that if you have a single molecule, it's going to be buffeted by all kinds of noise, whether it's thermal noise or the noise of quantum mechanics, basically chemical reactions. How could it possibly be that A, it is not changing over time, so inheritance is so sharp, and B, how can it contain information? And he came up with this concept of an aperiodic crystal, which is a very wonderful phrase. And this book and this phrase actually inspired the discoverers of DNA, Crick and Watson, to look for something of that kind in their crystallographic models. Okay, so that was Schrödinger. So now I'm going to jump back, okay, because I want to give you the history of how DNA was actually discovered. It wasn't just suddenly that people found the crystal structure of DNA and then everything was explained. Okay, it was a bit more complicated than that. So in the early 1900s, people were doing experiments with biology. Biology is an experimental field, and they were growing organisms, they were growing bacteria, they were growing other kinds of creatures, and they were doing biochemistry. They were isolating different types of molecules that are inside biological organisms. Okay? And one of the important contributions came early from Griffiths, who saw that a certain type of trait, so let's say the ability to resist a certain type of antibiotic, okay, that trait could be transferred from one organism to the other without actually putting the organisms together, just by transferring the dead molecules from one to the other. Okay, so again, this idea of particle of inheritance and it's being a molecule was already there. Okay, he didn't know which molecule. He basically took extracts. This extract is a mess. It contains everything that's inside a cell, but somehow in the extract there is the ability to confer a trait onto another organism. Okay, so after that there was a lot of discussion. What is this molecule? Are there one molecule, many molecules, and so on? What you had to do is to explain the following ability of biology. As Spencer said, biology, the crucial thing is the ability to clone itself, right? So you have a cell. Again, let's think of bacterial cells to keep it simple. A bacterial cell grows and divides. And once it's divided, you have two identical bacterial cells, essentially. Okay? These two are the same size as the parent because they've eaten food and grown. And they also contain exactly the same capacities to metabolize, to move, and so on and so forth. Okay? So, of course, you all know about proteins, so I'm going to cut to the chase. All the business end of biology is done by proteins. These are molecules which are very complicated, three-dimensional objects, okay? Several nanometers in size. They lock together, they can have force generation, they can bind to other atoms, and so on and so forth. And proteins are the things that, for example, enzymes are proteins. Okay? So one theory, which is completely plausible, is that every protein in the cell acts as a template for a new protein. Every protein makes a copy of itself. So then it's easy, right? Then you can just make a copy of the whole organism by copying all the proteins. That's one theory. It seems a bit far-fetched that a protein could make a copy of itself without all kinds of other machinery, but it's completely plausible. Okay? Then, of course, there was other stuff in the cell. And the way people like to think about it is, so if you look at the atoms that you find inside organisms, of course, there's oxygen, hydrogen, carbon, and so on. But then there's two other important ones, nitrogen and phosphorus. Okay, nitrogen is the stuff of proteins. They're made up of amino acids. And phosphorus is the stuff of DNA and RNA. They're made up of what's called the sugar phosphate backbone. Okay? So it's actually quite easy to separate these two because we had radioactivity. So you can actually have radiolabeled nitrogen and radiolabeled phosphorus and thereby isolate these two subsets of molecules inside a cell. Okay, so I'm going to explain this little slide here. It's a bit complicated, so I'm going to go through it very, very slowly. This is a bacterium. It's actually E. coli, which is the bacterium we work with in the lab. This thing here, which looks like some sort of satellite or a spacecraft, it actually looks like that, is a bacteriophage. It's a virus that infects a bacteria. So bacteria also catch colds. Okay? It's well known that the virus will bind, it was well known even at that time, that the virus binds to bacteria and somehow transfers material from inside here to sort of storage compartment to inside here, the cytoplasm of the bacterium. Okay? So, and once that's done, the bacterium actually has new traits. It's able to do different things. Right? So the main question, if you had to answer this sort of ambiguity, is it DNA or proteins, you just had to ask, is it nitrogen label that's being transferred into the cell, or is it the phosphate label that's being transferred into the cell, or the nitrogen label? The nitrogen labels proteins, the phosphate labels DNA. And it was very, very clear that the nitrogen just sticks around with the virus, which then gets washed away. But the phosphate stays inside the cell. And this basically clinched it. It was DNA that was this molecule, which was previously an abstract particle of inheritance. Still didn't explain anything. How is it that a molecule can have all these complicated properties? And that's why we have Watson and Crick and Rosalind Franklin discover, through a lot of work, the structure of DNA. And you should understand from what I've said so far that this could not have come too soon. There were a huge number of ideas about how could it possibly be that DNA contains information? What is the structure of this molecule? How is it that you can write something in there? It almost looks like a book where you can write something. So it turns out DNA looks like this. I know you've all seen it, but let me just go through it in great detail. DNA is a double helix. There are two independent strands there. Okay? It's actually a right-handed double helix. If you point in the direction, then the right hand curls in the direction of the twist. Right? So it turns out that, including on the, I think the web page of the ICTS till recently, they use left-handed DNA. But this is not, so there's a lot of left-handed DNA going around in publications and so on. DNA is right-handed. It's also anti-parallel, and this is a sort of subtle point. This molecule has an orientation. Okay? It points in one direction. The other molecule runs in the other direction. Okay? So overall, the double strand of DNA has no orientation. Okay? It's an anti-parallel double helix. Good. What holds these two pieces together? Well, that's these little sort of ladder segments that connect the two helices. And if you look at the labels, they sort of look small over there, but the labels have 4 characters, A, T, G, and C. You've all heard this before, but it's worth reiterating. There's only 4 types of chemical varieties that occur inside that double helix. Only these 4. Could have been many others, but it's only these 4. They also have a very important and interesting property. You can't quite see it here, but basically by shape complementarity and by hydrogen bonding, A likes to stick to T and G likes to stick to C. Right? That's the best configuration for them. So this, the first part of it, the double helix, shows something important. It says that actually DNA does look like a book. Okay? It's a long blank sheet on which you can write things. Okay? This is Schrodinger's aperiodic crystal. How do you write things? Well, you write things using just 4 letters. Right? If this seems odd, remember the computers write things using just 2 letters. Right? So not complicated at all. So that sort of explains how DNA can contain and store information. This, which is the Messelson and Stahl experiment, very, very important again, shows the other piece of the puzzle. How is it that you can make a copy of a bacterial cell that are identical, right? So the trick is the following thing. You don't make copies of all the proteins. You make a copy of the DNA itself. And how do you make a copy? It's because of this A to T, G to C complementarity. Right? So what you do is you pull the 2 strands apart, pull them apart, and then you can just read the letters here and find their matching strand. And you'll realize that once I've pulled it apart and added the matching letters on the other side, I get 2 copies of DNA which are identical to the first one. Right? So DNA not only contains information, but it can make copies of itself, which is absolutely remarkable. The third thing, which is the piece of Darwin's theory, because if this was happening in a chemical reactor in the lab here, there would be a lot of mistakes going on, okay? Because chemistry is noisy. Turns out that DNA is surrounded by a huge number of complicated proteins that make sure that this process happens with extreme fidelity. Right? We're talking about sort of 1 in a billion type of mistakes. Right? So mostly information is inherited, however, there are small changes. Right? And you realize now that's all you need as the stuff of Darwin's evolutionary theory. This is the molecular side of Darwin's evolutionary hypothesis. Yeah, so where do these things come from? So these are just organic molecules, right? They're nucleotides which are connected to the sugar phosphate backbone. You can actually make today in the lab other molecules which are not these, and you can make DNA that contains other variants and with shape complementarity. Okay, so it's not that it's chemically impossible. Okay, but I'll get back to that question in the little interlude. Just please remember that. Okay, so I said that you can write in this book. right? And you can make copies of the book. That still doesn't explain what the language is. And that's part of what many people are going to do in the school, is try and tell you, if I have a DNA sequence, what is the meaning of what is written. It's like having a book in a foreign language and you have no idea what it means. You can clearly see that something interesting is going on, but what? So remember what I said. I said the business end of a cell, the stuff that does things, is proteins. Okay? And proteins are made up of these things called amino acids. And it turns out that proteins, also a bit like DNA, are also made of building blocks that are stretched into a long polymer. Okay? So similar to DNA, a protein can also be described as a chain of discrete building blocks. And it turns out the building blocks of proteins occur in about 20 variants. Those are called the amino acids. Okay? And this is another magical thing which we'll hear about later. If I put the right stretch of amino acids together and I put a protein in a solution which has the right sort of osmotic properties and so on, that long chain will fold into a shape which has the right electromagnetic charges on the surface, the right shape and size to do things. Okay? That transformation is what's called the protein folding problem, and we'll hear a little bit about it in the school. But let's assume that given a protein with the right sequence, it'll make the right three-dimensional structure. How is this sequence to be defined? Right? This is like you're in a factory and you need a blueprint to make a machine. The blueprint is actually in the DNA. And now I don't have to tell you, you all probably know. If you want to encode 20 amino acids using an alphabet of just 4 letters, right, you probably have to do it in triplets of letters. Right? So 3 letters of DNA. So here it says U, C, A, G, but the U is like T. Okay? You can ignore this. Turns out that in RNA, there's a slightly different chemical variant which is U, not T, but ignore that. If I take 3 letters on DNA, then I can make 64 possible combinations. And this thing is the dictionary. You'll actually find it in the back of every cell biology book and molecular biology book. Just flip to the back page, and some people have memorized it. This is called the genetic code. Right? It says that these letters correspond to that amino acid. So if I see any of these triplets here, then I have to put tryptophan, and so on and so forth. Right? So this is starting to explain the language of DNA. Right? Again, Nobel Prize winning work by Hargobind Khorana and others. It's digital information. Okay? So now I want to bring in the other sort of revolution that was happening in the mid-20th century, which you all know about. It's the information revolution. Yes? Are these letters completely equivalent? These 4 letters? No, they're not equivalent. Actually, some of these are more preferred than others. Okay, so it turns out if you use a less preferred one, it takes longer to make the protein. It's called codon bias. And you and bacteria have different biases of codons. But one important thing, this dictionary is true for bacteria and for you and for plants and everything else. Okay? So the 4 letters are the same for everybody. The dictionary is the same for everybody. Just keep this in mind. I want to discuss it later. Okay, so you all know who these are. Alan Turing, Claude Shannon, the giants of the 20th century, and really revolutionized how the whole world is today. Right, so it was sort of a pure coincidence that the science of information came around at the right time to understand what was going on in biology. It needn't have been, perhaps, that biology is sort of digital. But it turns out that it is. And therefore, the language that was invented by these people, the language of programs, okay, universal programs, it's called the universal computation, the language of information theory to quantify how much information there is. Right? These things were invented completely independent of biology. But it turns out that they're very, very applicable. And if you want to read more about this, I would direct you to Sidney Brenner's recent papers on this. You can just Google it, and there's plenty of thought on this. Okay, so what Shannon particularly allowed us to do was to quantify information. So although we don't really understand the language, we can actually quantify things. So here, you all know that digital information, these days everybody knows about it. It's measured in bytes, which is actually the wrong unit. So a byte is 1 letter of the ASCII code. Everybody knows this. The byte turns out to actually be 8 bits. Okay, so the real unit of information, the 0 or 1 question, is roughly 1/10 of a byte. Okay, but I'm going to span orders of magnitude here. A letter is 1 byte. A word is approximately 10 bytes. Printed page is approximately 1,000 letters. Huge jump over here. Pictures are worth, you can see how many words. All the works of Shakespeare can be written as text. It's a lot of stuff. Songs, digital music turn out to again contain many more bytes. It takes a lot of information to compress music, and so on. DVDs. And this is a nice thing to end with. The current size of the US Library of Congress is 10 terabytes. You can actually buy hard drives of terabytes in the market today and store all the information in the Library of Congress. Okay, so I've just left this over here. So I want to ask you, how much information in a single gene? Right? So if you had to take all the information that you need to encode a protein, how many bytes do you need? So this is where we start to get interactive. So somebody can, doesn't matter if you're wrong, but where I'm going to start going down, and if you think I'm right, then you can just stop me and say yes. If you know the answer, of course, don't. How much information in a gene? Just tell me when to stop. Oh, by now we've gone too far, right? Yeah, I'm going to go up again. Anybody? Stop here? Okay, somewhere between a song and the works of Shakespeare? No, it's just here. Right? Surprisingly, maybe not surprisingly, because information is exponential in the length of the message, right? So you can actually encode the entire sequence of a protein using a few number of bytes. So it turns out that proteins have on the order of hundreds or 1,000 amino acids, and each amino acid needs about 3 letters of, it needs exactly 3 letters of DNA, right? So 3,000 letters. So there we go.
Is there in nature a tussle between trying to compress information and trying to keep it fidelity? Is that what decides what?
Yeah, good. Maybe we can have a tutorial on this. The fact is that biological information is stored in a highly redundant, extremely robust way. It is not stored as highly compressed data. It's stored as data that is easily readable, right? Which is part of the reason why people are saying if you want to store information for the millennia, put it in DNA, because you'll always have the reader for it. Okay, so it's exactly not compressed. Yeah, because you don't need to compress it, because DNA is a small molecule and you can fit it inside a cell. But that's not really true. Okay, so now you have a gene. How much information? So if I want to make a blueprint for a bacterial cell, which is the simplest living organism, right? Many people agree on that statement. How much information do you need to make a bacterial cell? I'm going to start. Obviously, it's more than a gene. So stop. Wow. Okay. Yeah. Okay. 1 megabyte. So it's nothing, right? So the amount of information you need to encode an entire bacterial cell is only this much. The way to think about this is the following thing: how many different types of things could you encode with this many letters? It's a very large number. Out of all those, okay, one of them is a bacterial cell, and the rest of it is nothing. Yeah, the rest of it won't make anything living. Bacterial cells comprise a very, very small part of the whole informational space. How did that information even get discovered? It's through evolution. Evolution has searched the space and found the thing that makes a cell. Okay, so obviously the next question. So human, right? So the bacterium is here, and for those of you who know the answer, don't answer. But for the rest of you, just take a guess. So where do we stop here? Stop. Here? Up? Anybody up? Up? Okay. Oh, so now you're saying, oh, it's very. Well, yeah, you were right the first time. Okay. Look, it's not much more than a CD, and it's certainly much less than a DVD. Right? So when we talk about personalized genomics, for example, your entire DNA sequence can be compressed and encoded and given to you in a thumb drive. It's really not much. So one of the big, in terms of information storage, right? But it's certainly a lot in terms of trying to understand what's written in a book of this size. Right? Certainly much more than all the works of Shakespeare and comparable to all the works of Beethoven. We need to understand this language. And the language of DNA goes far beyond the language of genes, which I just explained to you. Right? The genetic code, which explains how you take DNA and make proteins, is just the tip of the informational iceberg. And a lot of the work that's going on today is to read the rest of the genome, which doesn't encode proteins, to understand what they're doing. Okay? Now, I've been talking about reading genes. We can actually manipulate DNA. Right? And this is something that we've learned by borrowing, again, proteins from living organisms. So 1973, something called cloning was invented. It's basically the ability to take DNA from one organism to cut out in a very precise sequence-dependent way, exactly from one point to another, make small fragments, and insert those fragments into another target piece of DNA. Right? So this is called recombinant DNA technology. You might have heard about it. And it gave people, for the first time, the ability to take genetic material from one creature and put it into another without going through this process of breeding. Okay? Now, it also gave people the ability to take DNA from humans and put them into plants or bacteria, right? For which no amount of breeding would ever work. Yeah? So this is very important. Once you've made DNA, another important advance, and this is really driving a huge part of this revolution we've heard about today, is sequencing. I have a DNA sequence. Now, what does DNA look like? If I put DNA in a buffer in a tube and I give it to you, in fact, I can buy DNA in the open market. They'll send me DNA. It looks like a clear liquid. Okay, it might be viscous and has certain other properties, right? But it's a clear liquid. What is the sequence on the DNA? So sequencing actually allowed us to read the sequence of DNA. I give you a single DNA molecule, rather I give you a large number of identical DNA molecules, and you can actually read the letters written in the book. Okay, and that was Sanger sequencing, and Sanger actually passed away just a short time ago. Huge revolution. Now, the next thing, I said you need large numbers of identical copies of DNA to make this work, because this is happening in a chemistry lab, whereas DNA is a single molecule. But how do you transcend those two scales? That was this amazing discovery of on a beach, I've heard somewhere, called PCR, Nobel Prize winning discovery, which allowed DNA to be copied with extremely high fidelity in an exponentially increasing fashion. Right? So with 10 cycles of this, 1,000 copies of DNA. 20 cycles of this, 1,000,000 copies of the original strand. Right? Fine. Today, actually you saw a slide of this in Matteo's introduction, the cost of sequencing and the cost of synthesis have just crashed. So in fact, this is a machine which contains several bottles. Each one of these bottles contains a chemical. There's A in there, T, G, and C. Okay, that's literally how it works. It's connected to a computer. You type in your DNA sequence that you want in the computer, right? And this thing will make the DNA for you. It's a little difficult to make very long strands, but we'll get there. Right? So the fact is we can just make DNA. Therefore, we can create genes that are not even out there. Just make them. Recently, there was this on the cover of The Economist. It says, "A man-made life." So you might think I'm just leading up to the point where I'm saying that we have so much power that we can make living things. So this is the cover of a popular magazine, and it's actually completely misleading. This is the first artificial organism and its consequences. This is completely wrong. Okay, let me just put that out there. The actual paper, if you read it, says creation of a bacterial cell, even this is sort of wrong, controlled by a chemically synthesized genome. So what they did here, this is Craig Venter and his entire team. It's a huge advance. I'm not saying it isn't, but it's not the creation of life. What was done here is, like I said, you can type DNA sequence and make it in a machine, right? So they actually found a way outside of a cell to make a long stretch of DNA. But even that, actually, they had to use cells. But let's assume that they made a long stretch of DNA outside cells. Then what they did was put that DNA into an existing living bacterial cell. And just like Griffiths did in the early 1900s, they transformed the behavior. Right? That we've been doing for a long time. Here it was done by a chemically synthesized genome. So you can ask, is that really artificial life? Well, then you have to understand what it means. What is information? What is process? Right? So I've told you that DNA is purely an informational substrate. You could think of it like a CD or a DVD. That's all it is. Right? If I gave you a CD and you didn't have a CD player, that is no use at all. Okay? The DNA can only be read out if you have the reader. The reader is a cell. We can't make cells from scratch, at least not yet. If you are able to read, then you can make the final product. Right? In the same way, you need the computer or the DVD player or whatever it is to read the information on a digital format. So saying that I invented this, I made a new CD, excuse me, is not really the same as saying that I've invented life. Not even close. Another way to think about it. Right? It's very easy to copy a work of art, but it's much harder to actually make the work of art originally. Right? So we have not even come close to inventing life. We certainly have a lot of power to manipulate life. Okay? So this is where I'm going to transition into the rest of the talk, which should take about 45 minutes. But I just want to pause over here and ask you all to contemplate the following thing. Why is it? So if you look at now everything I've described, then life on Earth, there are sort of three big ideas. Okay? So this is borrowed from Paul Nurse's idea for big ideas in biology. Evolution is a big idea of biology, and it explains a huge amount of stuff out there. Okay? It's universal. Everything on Earth that is alive evolved, is a product of evolution. Okay. The other thing, which is universal, and I'll talk about it tomorrow, is that everything alive is made up of cells. Unexpected, but it's a fact. The third thing is, everything alive stores its information in DNA. There are three universal facts about life on Earth. Can you think of some reason why some of these universal facts are sort of less equal than others? So DNA, cells, and evolution has come up yet with an alternative to explain how complexity can be ramped up. This is Dawkins' entire thesis. What about DNA? There's no reason why life on other planets should be made up of DNA. Any informational substrate will work. What about cells? This is on the boundary. Okay, it's not at all proven that you need cells to make living things. On the other hand, cells are a very, very interesting way to get stuff done. And if you think about early molecular evolution, right, Albert Lipschaber is doing work on trying to make cells from scratch. So it turns out that cells are very important for DNA to keep its information private. Right? So evolution may not even get started without cells. So the jury is still out there. Certainly not DNA, it might be cells. It almost certainly is evolution. Okay, that's one fact. Second fact, why is it, and you've got to think about this every day of your life, why is it that the dictionary in bacteria is the same as the dictionary in humans? There are two reasons for this. The same origin, right? So as far as we know, life arose on Earth once, or at least all living things today came from the same origin of life, right? So therefore we all use the same language, not a problem, okay? But that was 3.5 billion years ago, right? During that time, the speed of light has changed, the distance of the moon to the Earth has changed, and so on and so forth, right? So galaxies have moved far apart from each other. Why is the language the same? Protection? Okay, interesting. Robustness? You mean this is the only way we could have written a language? Yeah. So this is sort of an optimal language that might work for life, right? But if, yeah. Okay. So the idea is that it's hard to change a language once you're already working with it, right? But actually, if you look at human languages, which linguists also borrow ideas from evolutionary biology, they describe the growth of languages on a tree. Not quite, but human languages do change over time. It turns out to be completely viable. In fact, we can make synthetic organisms today that have a different language. It's very easy to do in the lab. And if it's easy to do in the lab, then surely over billions of years it's easy to do. Fine. So it's a sort of rare event argument. Yes. Good. That's the answer. Okay. So in ways that we're only beginning to understand today, okay, my genome is not my own. Okay. DNA from all across life, from bacteria to plants, the things we eat, the things on my skin and so on, are moving around between different cells. Okay. So an amazing fact that I read about recently, so a woman who's had a male child, there are cells in her body which will contain the Y chromosome, which women do not have. So it came from the father into the mother. Right? These are sort of in small quantities, but they're there. Right? My genome contains traces, large number of carcasses of viruses that are sitting inside me. In fact, viruses are the single biggest carrier of DNA between different organisms. They'll cut out DNA from one place and they put DNA in another place. Our ability to cut and paste DNA was borrowed from viruses. Right? So DNA is moving around, and somehow biology is more fruitful if all the information can be shared. Right? And so an organism that's using a completely different language just cuts itself off from all the progress that's happening in the rest of biology. And therefore, maybe in the game of natural selection, will not persist. I'm not saying this is proven, but you need something to explain the striking fact of conservation of the language over 3.5 billion years. So you can think about that as the school proceeds. Yes? Yeah. So it's possible that an organism that uses a completely different language is resistant to attack from viruses, right? It's only resistant to informational attack. It's completely resistant to physical enzymatic attack, right? So as a way to hide, I can change all the language in my house, but somebody will come and burn the house down, and that doesn't really help me. Right? So it turns out to be better for biology to share your progress than to hoard it, right? And we'll talk about this later during the evolutionary sections of the school. Okay, so I have a lot to get through, and I'm going to change gears here. If we don't finish this section in the allotted time, it's not a problem. We'll continue in the period after lunch. Okay, so don't get too disheartened. Change gears completely. Okay, so I did mention that a lot of the information in DNA is not just to make proteins. It's something else. So what I'm going to describe to you is what all the other stuff in the genome is doing, at least a lot of it. Okay, so let me start with this. This is E. coli. It's an electron micrograph of E. coli. Size is approximately the wavelength of visible light. This is the most well-studied living genome because biologists chose to study it in the early 20th century. Now, E. coli contains 4,000 genes. That means it contains information to make 4,000 different proteins. Okay? And you all saw in my little chart of bits and bytes, the human genome is only a factor of sort of 6 larger than that. Okay? This is also striking. Maybe not unexpected if you thought about it. So when I first show this slide, a lot of people get taken aback and saying, so we're not 6 times as complicated as a bacterial cell. And I agree with you. Right? Of course we're not. We're much more complicated. We don't know what the number is, but it's certainly not 6 times. This is a misleading reading of these numbers. So it turns out that E. coli has 4,000 genes. It can make 4,000 different proteins. But if you take a single E. coli cell and manage to take all the proteins out inside, it would have only been making about 1,000 or less of all the proteins it could make. Because, for example, it doesn't need to metabolize glucose when there's no glucose. So it doesn't make the glucose metabolism enzyme. Right? In other words, it can choose to turn on or off every one of these genes. Right? So now, if we just extrapolate naively, there's sort of 2 to the 4,000 states that a bacterial cell could be in. Obviously, it's not true, but my point is this should be in the exponent. Right? And if you put this in the exponent, and now you can see where the complexity comes from. Right? Complexity in life comes from the fact, absolutely, that genes can be turned on and off. How does that work? This is important because a lot of things you'll see in the school will use the same language. So please try and remember some of this. If you don't remember, you can always ask people later. So this is DNA, double helix. I mentioned that there are certain bits of DNA which are on the order of 1,000 letters long, which contain the information to make a protein. Okay? Technically, this process actually goes through an intermediate called messenger RNA. Right? So you can think of this as a recipe book. You can think of this as the shorthand recipe you stick on your fridge, which you use every day, and that's the protein that you make. Okay? But it's basically DNA, genetic code, ATGC, triplets to proteins. But there's a lot of stuff between genes called intergenic region, which is small in bacteria, but it exists, and it's very, very large in humans. Most of human DNA is intergenic. It doesn't encode proteins. What that does is it turns genes on and off. Right? All this stuff here is a little molecular computer. It's interacting with the environment, and it's deciding, do I read this or not? Okay? It happens in a purely physical way. How does it happen? Well, a gene could be off. For some reason, it's not making the protein. How do you actually read it? Well, there's a molecule, it's a machine called RNA polymerase, a very complicated machine. It binds to a certain region in a sequence-dependent manner. Okay? And it actually moves in one direction. Remember, there is a certain orientation to DNA, right? So the RNA polymerase binds here and uses energy to move in one direction. Okay? And that's the reader. That's the hard drive reader, right? And that makes the protein. Actually makes the RNA, and then another machine similar to this called the ribosome makes the protein. The time scale, you can finish reading the whole gene in the order of seconds, less than a minute. Right? But the time scale between independent binding of polymerases is again on the order of seconds, because one has to clear before the next one comes. They're big machines. Yeah? Okay. So the time scales here are minutes, if not longer. Okay? This is not happening rapidly. Right? You have to remember, it's happening in a molecular soup, which is completely messy. The very fact that this protein finds one stretch of DNA of maybe 10 letters long in the entire target space is something you have to explain. And it turns out that you can explain the time scale of minutes, seconds and minutes. Okay? So this gene, right, very good. So here I've shown an example of a gene where if the polymerase doesn't bind, it doesn't get read, but here the polymerase likes to bind. Okay? So in the absence of anything else, there is a physical affinity between these two, right? And therefore it will continue to read at a certain rate. It's a stochastic process, right? Okay. You can imagine somehow that this process can be perturbed by the presence or absence of other proteins. That's basically to answer your question, right? So in this case, you had a gene which was originally off, remember? Right? It's off. How do you turn it on? Well, suppose there's another protein here, which is a green light. It's like a green traffic light. It's called an activator. That also binds to a certain sequence here. And that somehow increases the affinity of RNA polymerase for this region. Then only in the presence of that protein will this protein do anything. Conversely, in this case, the gene was on in the previous slide, and this slide the gene is off. Because RNA polymerase originally was binding, but somehow the binding of the red protein either occludes the binding site or it sticks to polymerase, prevents it from reading. There's any number of ways that could happen, but it stops it from reading. Okay? Now you could ask, where does this protein come from? I haven't explained anything. Well, that's easy. This protein comes from some other gene, right? And that might be regulated by some other gene, and so on. But it's not sort of turtles all the way down because this loop can close. Yeah? And this is why, as soon as you realize genes can be turned on and off by proteins, you have to immediately realize that means there are genetic networks, genes that connect to each other. So let's talk about networks, right? I'm just going to use a very, very simple abstraction for networks. If a gene is not making a protein, it's 0. If it is making a protein, it's 1. Right? You have to understand that when you unpack this abstraction, it means a lot about molecules binding, unbinding, and so on and so forth. Okay?
Is there a stochastic event that this many molecules of activator be present?
It can be. There's a lot of computation going on here. And you could have thresholds of the activator, you could have combinatorial effects of the activator, all sorts of interesting things could happen. Right? So this is basically doing computation to see what are all the other proteins there. Right? How, for example, does myosin, the muscle protein, only get expressed in muscle cells? Because muscle cells contain all the right combinations of proteins, large numbers of them, to say go. Right? Okay, so this arrow, sharp arrow, I use to describe activation. This blunt arrow I use to describe inhibition. And in this binary world, very easily, if it's an activator and the activator is not present, the gene is not turned on. If it is present, the gene is turned on. If it's an inhibitor, that means the inhibitor has to be absent for the gene to be on. It has to be present for the gene to be off. Right? This is only the tip of the combinatorial iceberg, but let's just start with these interactions and see what we can do. Well, turns out that you can make networks of these genes. You can take one gene, connect to another gene, and so on. Okay? So here's a few networks. I'm going to describe them in greater detail in the next few slides, but I just want to explain. Right? Here's a single gene that turns itself off. What does that do? Let's talk about it in a second. Here's a single gene that turns itself on. You can quickly realize that means if it was on to begin with, then it's happy. If it was off to begin with, it will stay off. So this is actually a bistable circuit. It is a memory element. Right? It's like your little flip-flop that stores information on your hard drive. Right? Similarly, if I have gene A turning off gene B and gene B turning off gene A, there are two stable states. Either the first one is on or the second one is on. Right? By the way, you don't have to take a binary model of this. Part of what you're going to learn in the school are other ways to model systems. And a lot of people use dynamical systems model to explain the same thing. In a dynamical systems model, you expand your model slightly to say a gene is not just off or on, but it could have a certain expression level, which is proportional to the protein expression level. And therefore, you have this phase space, and the trajectories move you around in phase space, and there are fixed points, and there are separatrices. Yes. Oh, sorry, this axis is the concentration of the protein made by the first gene, and that axis is the concentration of the protein made by the second gene at this instant of time in a two-state model, which is still great simplification. All I'm going to do here is take the two inhibitor system and make one activate, one inhibit. We'll discuss this extensively later. Turns out what this does, it makes an oscillator. It makes a biological watch. We talk about blind watchmaker, this is a watch. This is a famous experiment that basically kicked off the field of synthetic biology in 1999 by Michael Elowitz and Stan Leibler, where they took 3 repressors and put them in sequence, and they made another oscillator. And I'll describe this in great detail later. Okay, so here's what we do. Okay, fine. All these networks, in principle, using these very simple models, do interesting things, right? But that doesn't prove anything, because biology is messy, and if you really want to understand if it's possible, the best way to do it is actually try and make it. So it turns out that, like I said, to make DNA and sequence DNA is very easy these days. It's also extremely cheap, right? You can come to my lab for 2 weeks. I can teach you how to do it. So, why not try and make stuff? Okay, so here's what we do in the lab. This is E. coli, bacterium. It's completely passive. It doesn't cause infection, at least this species. It lives in your gut. Okay, that's the chassis. That's the framework within which I'm going to build things. Okay, here's DNA. There are certain bits of DNA where proteins bind. Those regions are called promoters. Okay, this sequence will determine whether an activating protein or inhibiting protein will bind over there. Okay, to match that, I need the protein itself. So those are called transcriptional regulators, because the process of making mRNA from DNA is called transcription. Okay, so the protein will bind to the promoter. And this example is a transcriptional regulator from a different bacterial species that lives in the ocean. Now, putting all this inside the cell is not enough as an experimentalist, because having everything inside doesn't allow you to control anything, which is a very bad situation to be in experimentally. So that's fine. It turns out that cells, because they have certain pumps on their surface, are permeable to certain small organic molecules. And these are just sort of small organic compounds, or even ions. Okay? So you can buy this off the shelf. You can buy it from a chemical company. They're called inducers. What they do is they come into the cell, and they can actually bind to proteins and change their behavior. I'll show you examples of this later. So I can control the cell. Furthermore, I can also see what's going on, right? Because among the many proteins I can put inside the cell, I can put proteins that are fluorescent in many different colors: blue, green, red, violet. Yeah? This example is called GFP, the green fluorescent protein, and it comes from the jellyfish. Okay? And this is a Nobel Prize winning protein, as it were. So this is what we can do. And again, you have to worry, how is it that I'm able to take a viral promoter, a bacterial transcription factor, a jellyfish gene, put them inside the cell, everything works? That's because of the single origin of life and the conservation of the language. So before you think it's too easy, getting things to work is hard. Okay? So when I moved to Bangalore, one of the first things I did was, well, we have this lab and let's have a summer school where we called a lot of students, both from NCBS and outside, and said, just think of what you want to make. I have the money. We're going to synthesize DNA. Let's just make it. Okay? So the students came up with 3 big projects. Okay? One was to teach cells to move chemotactically on 2 different axes. The other one was to teach cells to synchronize their cell cycles. And the third one was to teach cells to report DNA damage in their past. It doesn't matter what they were. Point is, we tried all this over a summer. It's not that we made the DNA sequence wrong. We put it into cells, but in practice, the design did not function as expected. We were not able to restore chemotaxis. Synchronization could not be achieved. Right? These statements are actually from a paper which contains nothing except 3 negative results, which is published in IET Synthetic Biology. So I was actually quite happy with this, right? Because rarely do you get to publish negative results. And some of you already know the punchline here. This totally negative result paper was actually cited, which is amazing. This is my greatest achievement. So people realized we did try our best to do something, and yet we failed, and therefore something needs to be learned. Okay? It's an example of science actually working. But we need to make sure that things don't fail. So let me explain how that works. These networks which you saw on the previous slides, these are all examples of networks people have actually built and they worked. This is not my work. I'm just giving you a survey of the field. Okay? So Attila Beskey, Luis Serrano, Nature 2000. Again, that 99-2000 was the year that all this got kicked off. And part of the reason why I got excited by this. This is called negative feedback. Okay? Negative feedback means you have a gene. In this case, it's called a tetracycline repressor. It represses the tetracycline promoter. Okay? And also because we need to see what's going on, we put GFP in front of it. Okay? And in this particular experiment, what they did was they made 2 different types of bacteria. One where the feedback did not work because there was a mutation in this protein. The other one where everything is working. Then you put this DNA into a bacterial cell. You let the cells grow and divide. You have a bunch of bacterial cells. You put them all under a microscope slide and you measure how much GFP there is. And what you see is when negative feedback is working, it's a very, very sharp expression of GFP. That is, this is a histogram. These are very bright cells. These are very dim cells. There aren't any very bright cells. There aren't any very dim cells. Most of the cells are close to the average. If you kill the negative feedback, this becomes very broad. Right? So this is a well-known motif from engineering. Negative feedback reduces noise. Because if a protein expression level goes above its set point, its synthesis level drops. And you come back to the set point. So this is an example of a success story. Single negative feedback. Here's an example of another success story. Again, 2000, months, one after the other. 2 repressors. Okay? In this case, what you have is this gene, which is called the lambda repressor, which is actually called C1, okay, which represses the lambda promoter. Then you have the lac repressor called lac-I, which represses the lac promoter. Okay? So, and then, of course, you put GFP on one side so you can see what's going on. And these promoters point in opposite directions. It can happen, and they just chose to do it this way. It's not necessary at all. Here's an important thing. They actually had a way to control the cell from the outside. If you add heat, this protein would degrade, right? And therefore, this repressor would no longer work. Therefore, this protein synthesis level would go up. And conversely, if you add this small molecule called IPTG, this direction would no longer work. So this expression would go up. So here's the result. Okay? I start off with no GFP. That means all transcription is happening in that direction. Right? I add IPTG. That means GFP level is going up because this negative feedback is not working. Then I remove IPTG. I go back to the original state, but the cells still stay in a state of high GFP. And why is that? That's because not only have you made GFP, you've made this repressor, which now keeps this silent, whether or not there's IPTG. Right? In the same way, you add heat, you come down, you remove heat, you stay down. So this is a standard classic bistable switch. So here's something I did for my PhD. This is a long time ago now. So this is a single positive feedback, which also turns out to be a bistable system. In this case, the way it's done is E. coli cells naturally have a system of a protein called lac-Y, which is a pump. Remember I said there are pumps in the cell surface which can pull in a sugar called TMG. And that sugar, through a series of interactions, increases transcription at the lac promoter. So this is a single positive feedback. And to see what's going on, we put GFP downstream of the same promoter. You put this construct inside a bacterial cell. You put the cells on the microscope slide. This is what you see. This is a pseudo-color image, but quantitative, showing that some cells are extremely bright in GFP, some cells are extremely dim. Okay, cool. And this is not the only thing you can do, right? Because you can change the external sugar concentration. So let me show you the whole experiment. Right? This is a summary of the whole paper. X-axis is the extracellular sugar concentration. Y-axis is actually the green fluorescence on the log scale. A single experiment corresponds to a single column over here. Right? So this experiment was done at sort of 15 micromolar TMG. A single dot is a single bacterial cell. So what you see here is because there are bright cells and dim cells, there's sort of a cloud which is bright and a cloud that is dim with a factor of 100 difference between them. Now, you heard the word hysteresis earlier in Rahul's talk. So basically you increase the level of TMG and eventually the cells flip to the on state. But you decrease the level of TMG and at a much lower TMG concentration they flip to the off state. So this is, again, think of external magnetic field, magnetization, this is hysteresis, right? And the mathematics is entirely the same. Right? So this is work that was done with Boris, who was sitting in the audience. It was a lot of fun trying to make the connection between physics and biology in this case. But one important thing, if I take the middle concentration of TMG and I tell the student, I've done this experiment at, say, 7 micromolar TMG, are the cells off or on? She won't know because it depends whether they started off or started on. Right? So it's a memory circuit. Very quickly, this is the famous repressolator. Lambda repressor represses lambda promoter, lac repressor represses lac promoter, tet repressor represses tet promoter. You should all be very familiar with all this jargon by now. And then you put GFP downstream of tet promoter to see what's going on. 3 negative feedbacks have no consistent steady state, and therefore the system will end up oscillating. Right? This is experimental. Sure. So I'll tell that. That's exactly what the rest of the talk is about. So remember, so far the school is about quantitative biology, right? So I'm just trying to make the point that the idea of quantitating an experiment is important. It's not so much how sophisticated your model is, but it's the idea to bring a quantitative mindset to the design of experiments. Here is a more complicated oscillator. I'm just going to throw it out here, okay? This is the hysteretic oscillator. A activates B, B represses A. This is an important example of an oscillator. It's found in all your bodies. I'll describe it later. Turns out to make it requires negative feedback, positive feedback, and also an internal positive feedback for various reasons. This is complicated. This is something we built in the lab. And as you can imagine, to build something of this type, you can't just do it from scratch and hope it's going to work. Right? It does work. Cut to the punchline, it is an oscillator, right? With a time scale of about 45 minutes. Okay? But how do you make something like that? That's what the rest of this talk is about. In the next half an hour, I'm going to tell you. Remember, I said when I first started the lab, things did not work. It's not because we didn't try hard, but because we get better at making things work. There is a process. Right? So what is the process? That's what I'm going to describe. So the game is clear. Right? I am the watchmaker's apprentice. I want to make a watch. By watch, I mean an oscillator. And I want to make a hysteretic oscillator because that's the game I'm playing. Okay? So I need proteins. I need to look at life and find proteins that will do what I need. In particular, for a hysteretic oscillator, I need activators and repressors and ways to mess with them from the outside. Right? So here we go. I'm just going to unpack the dynamics of the hysteretic oscillator. If both the genes are off initially, right, since there's no repression going on, the first gene will turn on. Right? Now that you have this gene is an activator, it will pull the second gene on. Right? Now that this repressor is working, it will shut the first gene down. And now that there's no activator, the second gene also falls down, and you keep going in circles. Same thing explained on a phase plane, where this is the concentration of the activator, that's the concentration of the inhibitor. These are the null lines for people who know the language of dynamical systems, and you get a limit cycle oscillation under certain parametric conditions. The reason this is interesting and important is because, as I said, this oscillator is found in every one of the cells in your body. This is the oscillator that's giving Matteo jet lag and maybe some others. This is your circadian clock. Right? This is what controls your 24-hour cycle. It's made essentially at its core with just 2 genes. Right? There's an inhibition and an activation. There's a lot of other stuff here, but that's the core of it. Right? Not only is it found there, it's also found to generate spatial patterns by having an oscillatory signal and a moving, growing tissue. So you can convert a temporal oscillation to spatial waves to make an animal. So my point only is that this oscillator is important in real life. Many people have also made this oscillator synthetically, the same one that I'm talking about. Right? The hysteretic oscillator has been made in many different contexts, most recently by synchronizing different cells by Jeff Hasty. So these things all work, but they're difficult to get working, so I'm going to tell you how we made ours work. This is the oscillator. It's very complicated, right? So essentially, there is a negative feedback here with the lac repressor, and there's a positive feedback here with a protein called lux. Okay? So initially, you have a lot of blue, very little green. Eventually, you have none of both. Remember the same 0, 0, 0, 1, 1, 0, 1, 1. The blue shuts down because the activator is no longer present. The repressor is there, so it shuts down the green. You go back to this, and you keep on circling. This is the oscillator I want to make. It looks horrible. But I can make it by thinking about the problem in incremental steps. So that's the point of the whole talk essentially, that if you want to make something complicated, use the ideas from engineering. Don't try and make this whole big thing. And this looks different from the previous slide for a very good reason. We realized that to make the big oscillator, you had to go through certain simpler stages which were achievable each. So it's only the incremental game we had to solve. So here's the game. What I need eventually is a system where this black box is the promoter. Remember what the promoter does is it executes a computation. It measures the level of proteins that are present upstream or in the rest of the cell, and it has an output which is the transcription rate. Okay? One of the outputs need to be a simple positive feedback. It turns out this is necessary to generate hysteretic oscillations. Otherwise, the symmetric fixed point is stable. The other feedback goes through the lag repressor. It's a negative feedback. Right? So once I drew it this way and not in this complicated way, right, this is horrible, this I can start talking about, because it turns out that this, imagine I cut that loop. Then it looks like this, which is a system with one input and one little feedback. Right? Imagine I cut this guy. Then I have this, which is a system with two inputs and one output. Right? So I'm trading the sort of characterization complexity for internal complexity. To characterize a system with two inputs and one output takes a lot of measurements. To characterize a system with one output, with one input and one output requires fewer measurements. This one has no inputs, so it just does what it's supposed to do. Right? So harder to characterize, but easier to understand. Okay? So this is the process I'm going to describe to you now. So let's start with what these proteins are. Yeah, question. So I'll address your question in a second. So he's an expert, so he knows the answer already. What are the pieces I'm borrowing? Here are the pieces. So it turns out that bacteria, this is what he described, it's a process called quorum sensing. Remember I said cells talk to each other, right? They all interact with each other. So it turns out bacteria have this property, which was discovered surprisingly recently, that in a population of cells, they actually chemically communicate with each other. Right? It was a surprising fact, and the way it was discovered was completely serendipitous. Turned out that there's one bacterial species called Vibrio harvii, which grows in a tube in the lab or in a flask, but in the dark, at low densities, you see nothing. But at sufficiently high cell densities, it turns bioluminescent. You can see it with the naked eye. Right? So bacteria have density-dependent gene expression. Okay, so a related organism called Vibrio fischeri, which we use in the lab, lives in the eye sac of a squid. And in the daytime, the squid flushes out the eye sac, and at night, the cells grow and divide, and at some point, the eye sac starts to become fluorescent for reasons we don't understand. Similar kind of game is played by organisms that attack you, in this case, Pseudomonas aeruginosa. It's a pathogen. It will line your mucous membranes, but it won't attack until there's enough of them. Now, why am I giving you this little segue onto density dependence? So it turns out density dependence is exactly the same as any communication protocol you can imagine. It involves two components, an issuer, which is called I, and a receiver, which is called R. Okay? And a signal, which can go outside one cell and come into another. So every cell issues a signal. It broadcasts a signal. You can think of it as shouting, or you can think of it as a chemical. The more cells there are, the stronger the signal. Every cell is also listening for the signal or smelling for the signal, right? So at some point you know, I can smell a lot of this, therefore I know there's a lot of guys, let's go. And you turn on gene expression. Right? So you only need two pieces to make this work, issuer and receiver, coupled by a signal. So these things turn out to be done by single proteins. That's what makes this a nice system to use. The issuer is a protein called I. It's an enzyme that makes a small organic molecule which can go between cells. The receiver is a protein called R, which will bind to this signal. And when it's bound to the signal, it will bind to this promoter called PR and generate transcription. So you have the issuer, the signal, which is AHL, it doesn't matter what it is, the receiver, and the binding site of the receiver. These are the pieces I use. Using just these pieces, I need to make a system which has two inputs and one output. Can I do it? It's very easy. Here's the promoter. Right? The promoter essentially binds the receiver, but it doesn't activate unless the issuer chemical is also bound. So some complicated stuff is going on in here. Right? But nevertheless, it's a two-input system. Issuer, receiver protein, black box, computation, and the transcriptional output. So I need to make this system inside a cell, right? And I did that. I put the issuer protein under control of the TET promoter, which you know about already, the receiver protein under control of the LAC promoter. So I was able to control both the knobs separately using small molecules. Right? So then what we did was, first of all, check that the knobs are working. So if you add ATC, you should have more of the issuer. How do we track this? Well, you label it with a blue fluorescent protein, right? Or a yellow fluorescent protein, or green fluorescent protein. So the first knob is working because I increase ATC, the issuer level goes up. As I increase IPTG, the receiver level goes up. So both my knobs are working. Finally, is the black box working? Well, yes, it is. That's a bit more complicated. I have to vary the blue input and the orange input over all possible combinations, which is a combinatorial experiment. So each one of these things is a whole day experiment done in triplicate at this particular concentration of ATC, that particular concentration of IPTG, and I measure the output. And after doing all this, right, this is 7 by 6 done in triplicate, huge number of experiments, but it can be done by robots. In this case, it was done by graduate students, but it can be done by robots. This is the hard part, right? And unfortunately, we have to cover this hurdle. We need the quantitative data. We get this, and this is what we call the promoter logic. Right? What that means is, as a function of all pairs of inputs, what is the output? So this is a three-dimensional entity, right? So this thing looks a bit like an AND gate. You could think of it like that, right? I need both the blue and the orange to make an output. Right? But not quite. It's not quite an AND gate. So this particular thing turns out, so this is sort of a quantitation of the same thing, right? We did the quantitation by making a biochemical model, using it to interpolate between all these points, inferring various parameters which had been independently measured, verifying that we had the correct parameter values, and then showing this in quantity. And you can see that it's not a symmetric function. Right? So it turns out that this particular model is sufficient to explain this, but that's not what I'm going for. My point is, this is the promoter logic, and that is the complete characterization of the 2 input, 1 output system. So the next thing I need to do is to go from the 2 inputs to the 1 input system. So I need, yeah, question. Oh, the proteins are always, all proteins are turning over. If a cell is green, that means that the balance between the creation of GFP and the decay of GFP keeps things at a constant level. So proteins are constantly turning over with a lifetime, well, in fact, proteins are being diluted as the cells grow and divide. Right? So that lifetime is about 20 to 40 minutes. We're measuring at a steady state. It's a steady state, right? So in this case, good question. Oscillators don't have steady states, but this thing has a steady state, so I can measure it. Okay, so now, second piece of the puzzle. I need a question. How do you decide the concentrations? Very good. Oh, we did. Yeah, so, I mean, these are all published previously, but if you didn't do it, you would just scan over log orders. And you would find that there's some region where things are changing, and then you would choose enough points here so that you correctly sample all the variation that's going on. Yeah, so very good question. Technically difficult, but it can be done. Yeah. Okay, so how to go from 2 inputs to 1 input? Mathematically, here's how you do it. Remember, here's a system with 2 free inputs and 1 output. Here's an example of a system where one of the inputs is coupled to the output, whereas the other input is left free to control from the outside. Here is sort of symmetric version of that. I've just flipped it around. Okay? Let me explain a little bit about what the mathematics of this is. Right? There was a question about how do you describe this. You need to write differential equations if that's what you want to do. dy dt, dyz dt means the rate of change of the concentration of protein Z. Right? This thing, to address the question in the back, contains 2 different components. One is the synthesis rate, the other is the decay rate, and it's the balance between these that leads to the steady state. The decay rate of proteins is typically a first-order kinetic process. Right? So it's a bit like radioactive decay. There's a time scale for that, which I've put on the left side. Right? Gamma Z. Okay. What about the synthesis rate? Well, the synthesis rate contains 2 components. One is the transcription rate, and the other is the translation rate, which, because there's 2 steps of gene expression in the central dogma. The translation rate is just a factor. The transcriptional rate is where all the complexity lies. Okay? It says what is the output as a function of 2 different numbers. Right? So it's some unknown function of both the input protein, of the issuer protein, and the receiver protein. And by the way, I'm just putting this in here, bacterial cell density also enters in as a product over there, because the issuer protein is generating the signal per cell. Right? But that's, you don't have to worry about that much. Anyway, overbar means fixed in time. Right? So in this case, I just keep the 2 inputs fixed. I measure the output, and therefore I can characterize the entire function f. Right? Modulo gamma z and qz. That doesn't matter. Once I have the entire function, which is what I had described to you previously, the dynamical system here contains exactly the same data, right? So dyr dt, which is the input in feedback, translation rate times transcription rate minus decay rate. The complexity is arising because in the transcription rate, one of the inputs is now a time-varying function. Right? This guy is the same as occurs on the left side. Therefore, this has to be solved a little more in a complicated way. Right? This is a system that could be bistable and so on. But it's the same function that occurs over here. And in fact, if I flip things around, I want to keep yr fixed and vary yi. That's the same function that occurs over here. So my point is, by measuring the 2 input, 1 output system, I can describe the behavior of both the feedback system. This is Oh, it's the same because the promoter is the same. That's the whole point. Yeah, that's the whole point. Okay, I'm just going to describe this to you slightly differently for those of you who weren't following the math here because I jumped through it. Here's how it works. I have a black box. The black box is my promoter, which is binding proteins according to some complex biochemistry and generating transcription at some rate. Completely complicated function in principle, right? My black box has two inputs. For the moment, just think of one of the inputs as being fixed, and let's vary the other input, right? So think of this as a fixed regulator. Think of this as the input I can increase, and I'm measuring the output. And what I'm characterizing here is just the input-output response of a black box, some sort of curve. Yeah, there'll be some curve, input versus output in steady state. Right? If I change the value of the regulator, maybe the behavior of the box changes in some unknown way, so you'll get some different curve. Fine. This is what I'm interested in. In this step of making an oscillator, I'm interested in going from 2 input, 1 output to 1 input in feedback. Right? This system and that system are actually very similar. In fact, they're identical in exactly the situation where the output happens to have a value numerically equal to the input. Because here the output and the input are identified. I equals O, not 1 equals 0. Okay, I equals O. So the output and the input are identified. So if I could find a value of the curve where the output was equal to the input, I can predict what this does. But that's easy. Just find out where this curve cuts the line y equals x. Right? And therefore, I can predict what this guy does. For a different value of the regulator, I can predict again what this guy does at a different fixed point. And therefore, this single input system, as I vary the regulator, which is now the input, I can describe the output. Right? I can talk a lot more about this, but I don't want to give you sort of too much to do. But the point is that this is exactly the step I need to do. I need to plot the input-output response of the single black box. I need to intersect that with the line y equals x. The intersection point is the predicted value of the feedback loop. Okay, so let's do that. This is in fact an entire paper, which I'm just going to cover very quickly over here. This is the promoter logic function. This is the behavior of the 2 input, 1 output system. Okay, this is the 2 input, 1 output system. Let us say I want to describe the behavior of this feedback system. In that case, I need to fix the blue guy. I need to vary the orange guy. Right? But I've already done that experiment. Right? That turns out to just be a vertical slice of this surface. Right? On the surface, I vary both the blue and the orange. So if I want to fix the blue and vary the orange, I just move up this way. Right? These data points I've shown, the black data points are exactly these data points. As I vary the orange, the green level is going up. These data points are exactly these data points. As I vary the orange, this is going up. This is the input-output function. Where it intersects the line, that's the predicted behavior of the feedback. This line is the prediction. It's not a fit. It's a 0-parameter prediction based on that guy. These orange dots are the actual behavior of the feedback system. Right? It's not a fit, and you see that we get it. Right? It's great. Actually, it's not, because this is a log-log curve, and these are all factors of 2 that you're off. So if you're happy with factors of 2, you count this as a success. If you're launching a rocket to Mars, it's not going to work. Okay, so in fact, this doesn't prove in a way that's really satisfying that we know how to predict behavior of feedback loops. But watch this. Serendipitously again, it turned out the other direction, where you keep the orange fixed and vary the blue, which are horizontal slices of this function, it turned out, because this function was asymmetric, that direction was much sharper. Right? And therefore, it intersected the line y equals x at 3 points. Which is actually the hallmark of a bistable system, 2 stable fixed points, 1 unstable fixed point, and the prediction was hysteresis as a function of the regulator. Right? So here's the predicted guy, not a fit. Here's the measured behavior. As you start with cells off, you have to go to a high level to turn them on. You start with the cells on. So anyway, point is it works. Not only does it work quantitatively, it works qualitatively. This seems a bit odd that qualitative predictions are stronger than quantitative ones, but you have to get used to thinking along those lines. Because there are many reasons why a quantitative prediction could be wrong. But there's very few reasons why a qualitative prediction should be wrong if your theory is correct. Fine. That was a lot of stuff, but this is the final thing. I want to go from the feedback system to the oscillations. What do we do? Well, take this system and connect the other side. Okay? Now, if you think about this in the language of dynamical systems, there are 2 dynamical variables here, the issuer and the receiver, which is on this axis and that axis. Okay? This curve is what happens if I vary this guy and measure the behavior of the feedback system. Right? That's what I predicted in the previous slide. That's the prediction. Okay? This guy is what happens if I vary this and measure the behavior of this in the absence of this feedback. Okay? I'm rushing through here, but the point is, Independent variable, dependent variable, I get this curve. Independent variable, dependent variable, I get this curve. They both intersect. And the prediction of the dynamical systems model is that if my degradation rates are high enough, I get a limit cycle. That's the limit cycle I'm supposed to get. This is what we actually get in the set. Right? So, to summarize everything, we didn't just make this from scratch. Right? We went through a lot of work, which took several years, to make sure that it was working, because that's how we chose the parameters. The experiments that you do is actually put the cells in a tube, you supply food, you remove cells, you add AHL from the outside to synchronize the cells, and then you can see that our oscillator in many different experiments actually provides sustained oscillations, synchronized across all the cells in the population over as long as we were able to measure them. This again was done by a graduate student, and therefore he was only able to run experiments for 40 hours straight, making measurements every 45 minutes. Right? Difficult. Right? Of course, now you could do it with the robot, but this is actually much more satisfying. Okay, so I'll finish. We talked about many things. So we're talking about timescales. You asked about timescales. How fast can you change whether a gene is turned on or off? Well, that's on the timescale of minutes, maybe hours. Okay, that's like the software of a system. You can change it fairly rapidly. Okay. You can also make mutations to proteins that will change the fundamental affinity constants. Right? That's going to happen over the timescale of thousands of bacterial generations, if not more. Right? So that is something which you can change. It's perfectly plausible, but you choose not to do it very often. Right? So that's a bit like your firmware. When you upgrade the firmware of your phone, you do it once a year. You don't do it every day. Finally, which guy is in feedback is a bit like the topology of the network, right? And that is actually very difficult to change. You change the topology, you can go from a smooth input-output to a hysteretic input-output. These are very drastic changes, which evolution finds a little difficult to deal with. So that happens very rarely. And so these are sort of the three axes along which things can change for these genetic networks anyway. So I'll stop over there. So this work was done initially by a lot of undergraduates and later by students in the lab. You can read more about it here. I'll be talking tomorrow anyway, and in the afternoon we can have more discussions about it, but you can also email me about this. So I think what I'm going to do is stop and take questions, and then we'll break for lunch. Thank you. Yeah.
You're modeling using differential equations.
Yeah.
The question would be that perhaps what is more natural would be difference equations. Discrete time.
I'll talk about such models tomorrow. So I will give an example where there are discrete in time, discrete in state models that apply to a different system. I should point out though, Eduardo Sontag and colleagues at Rutgers have shown that no matter how complicated the differential equation is in here, as long as it's described by a differential equation, right? Here this is a two-state system. Right? No matter how complicated it is, this particular procedure will work as long as it's a steady state. Right? So it's actually much more widely applicable than you might think. It covers all monotone input-output systems. So that's a good question. Yeah. Yeah, good question. Okay, so how do you vary the strength of the feedback? Well, I can do that in a few ways. One is I can actually add or subtract this coupling chemical independently from the outside. So that doesn't allow me to change the strength of the feedback, it allows me to shift the curve. Okay. The second thing is I can vary the translation rate of these things, here, here, and here. It's not a completely independent way to vary the feedback, but it can be done. Right? And luckily for us, it turned out that we were in a parameter region where we didn't have to vary all this, but we were completely prepared to vary it because we would know the factor change that I needed. Yeah? So that's a good question. So you've asked a very difficult question. So in fact, these systems are not differential equations of this type. They're actually sort of infinite dimensional differential equations because they have delay. Right? Binding of polymerase, the making of the protein, there's several steps of delay. Those delays turn out to fundamentally change the behavior of simple models, making oscillations where you don't expect oscillations in simple models and so on. And people are dealing with that. There are modeling frameworks and experiments that show that delay is important in some contexts, not in others. Good question. Question in the back and then here. Yeah. Okay. I urge you to take the Fourier transform of this. Those are all the data points we have, and that's the amount of variability that you get. Right? So you need either to sample much more finely, which is not really possible, or really to have 10,000 different replicates, which is not really possible. So taking a Fourier transform of this is not good. People, neuroscientists are lucky they can take Fourier transforms because they make measurements electronically. We can't do that. We make measurements optically. Yeah. Okay. Good. So thanks for your attention. This was a long session. This is the only time it's going to run like this. From tomorrow onwards, we'll have tea breaks galore. So let's break for lunch. Lunch is just outside, and we report back here at 2 o'clock for a very important session. Okay. So please be here.